Assalamu alaikum students. So today we will discuss the histology of tongue. Now tongue is a mass of striated muscles covered by mucosa which manipulates ingested material during mastication and swallowing. The muscle fibers are oriented in all directions, allowing a high level of mobility. Now, first of all, before going into the histological features of the tongue, we will see some of the gross features. Now here you can see that this you all know by your knowledge of gross anatomy in the region of head and neck that the tongue has got two surfaces, the ventral surface and the dorsal surface. Now shown in this picture is the ventral surface. This is the dorsal surface, the upper surface of the tongue, and you can see here that it has got an anterior 2/3 part. Here you can see the anterior 2/3 part and posterior 1/3 part here, posterior 1/3 part, and this anterior 2/3 and posterior 1/3 part is separated by a V-shaped sulcus, and in this V-shaped sulcus, in front of V-shaped sulcus lies these structures, rounded structures. So the anterior 2/3 of the tongue, as you can see here, this has got, the dorsal surface have got rough surface, rough areas, this surface is rough, while the posterior 1/3 is also rough. It is rather nodular. Posterior 1/3 of the tongue is nodular. So here you can see the posterior 1/3 and you can see that this surface is nodular. Okay, we will see what this section of the tongue contains later on. Okay, so this is the ventral surface and this is the dorsal surface seen here, and this is the ventral surface. So as you can see, the dorsal surface is rough while ventral surface is smooth and shining. So this dorsal surface is white and rough. This feature enables the tongue to roughness. This roughness of the dorsal surface of the tongue, this enables you to hold or grip the food particles when you are chewing it. You can do this by, you can assess this by putting some food particle below the tongue and try to chew it. You won't be able to do that. It will slip from side to side. But on the dorsal surface of the tongue, this roughness will grip the particles and you can easily chew them. So this is the dorsal surface and this is the ventral surface. Ventral surface is smooth and dorsal surface is rough. Now this is the histological section of the tongue on 10x power, and you can see here this section. This is the power usually used in the examination, and the section of the tongue appears like that, having the mucosa, the epithelium and the lining lamina propria, the epithelium and lining lamina propria, and then the muscle layer. So this is how it appears in the histological section. Now the dorsal surface, as I have told you, it has got roughness. It is irregular, having hundreds of small protruding papillas of various types on its anterior 2/3. The papillary surfaces, the papilla are elevations of mucous membrane that assumes various forms and functions. So you can see that this is the dorsal surface, and the dorsal surface, it contains papillas. These are the projections of epithelium. These papillas are projections of epithelium. So here you can see that the upper surface is smooth. This is the stratified squamous epithelium, and this is the underlying muscular tissue. This is the lamina propria, but you can see the surface is smooth. Here you can see the surface is smooth. No papillas, no projections of epithelium are present on the surface. So this is the ventral surface of the tongue. Okay, so as I have told you, the core of the tongue is lined by the muscles. So skeletal muscles are present. You can see here, these are the skeletal muscles with peripheral nuclei. This is the longitudinal section, while this is the cut section. And you can see the striations here in some of the parts of the muscle fibers, and these muscle fibers are running in longitudinal, vertical, horizontal direction. So this is the core of the tongue, consisting of skeletal muscle fibers running in various directions, and in between lies the connective tissue. Okay, now as I have told you, the dorsal surface of the tongue, it consists of papillas. There are 4 types of papillas present on the dorsal surface of the tongue. First of all, the filiform papillas, the filiform papillas, they are very numerous. They are the most numerous papillas in the dorsal surface of the tongue, and they have elongated conical shape. You can see here elongated conical shape, and they are heavily keratinized, which give their surface a grey or whitish appearance. They provide a rough surface that facilitates movement of food during chewing. Fungiform papillas are much less numerous and lightly keratinized and interspread among the filiform papillas. So fungiform papillas, they are mushroom shaped. You can see the dome shaped, mushroom shaped, and they are well vascularized, and they have got numerous taste buds on their apical surface. You can see the taste buds are present. These are the taste buds. They are present in their apical surface. Okay, then the foliate papillas. So foliate papillas here consist of several parallel ridges on each side of the tongue. So they are present on each side of the tongue near to the sulcus terminalis, but are rudimentary in human, especially the older individuals. In children they may be present, but in older individuals they are rudimentary. Then the velate papilla or the circumvallate papilla. You can see here velate papilla or circumvallate papilla. Now these are the largest papilla. Actually you can see it with your naked eye. You can see here the velate papilla, the rounded structures with diameter of about 1 to 3 mm. 8 to 12 velate papillas are normally aligned just in front of the sulcus terminalis. Ducts of several small serous salivary glands empty into the deep moat-like grooves surrounding each circumvallate papilla. I will show you here also shown, but I will show you in the histological section also. Now these are the large papillas, and they even can be seen with the naked eye. So these are the 4 types of papillas which are present, and the velate papillas have got highest number of taste buds present, and these are present along their sides. The taste buds in the foliate papillas are also present on the sides of the papilla, and in the circumvallate they are also present in the side of the papilla in the trench present around the circumvallate. This is the trench which is present around the circumvallate papilla. So here you can see that the taste buds are present on the apical surface of the fungiform papilla, and there are no taste buds present on the filiform papillas. So this is the histological section of the tongue. You can see here that it consists of an epithelium and then lamina propria and then the muscle bundles. You can see here. If you enlarge, you can see. Now this epithelium is stratified squamous non-keratinized epithelium, partially keratinized, because the filiform papillas, they contain keratin layer. Now this is partially keratinized epithelium, stratified squamous partially keratinized, stratified squamous partially keratinized epithelium. And then the lamina propria. In lamina propria you can see the connective tissue, the blood vessels, some of the nerves are present here, lymphatics are present here. So filiform papilla, you can see the filiform papilla, they are pointed structure, and they have got no taste buds in them. And this is the layer of the, you can see here, keratin layer, which is present on the surface of these filiform papillas. You can see here that the connective tissue of the lamina propria is tethering inside the muscle layer. It is going inside the muscle layer and tethering the mucosa to the underlying skeletal muscles. This is by you cannot separate your dorsum of tongue, the surface of your dorsum of tongue, from the underlying tissue. You can do it by yourself. It won't separate because the lamina propria tethered it with the underlying muscles. Now again, a picture of filiform papillas, and you can see the layer of keratinized epithelium here, and the underlying lamina propria and the muscles, skeletal muscles. Now this is the picture showing the fungiform papilla, and you can appreciate the taste buds present on the apical surface of these papillas. Apical surface of these papillas. Now this is the fungiform. This is the mushroom shaped papilla and fungiform, and they have got apical the taste buds on their apical surfaces. They are less numerous as compared to the fungiform papilla. Filiform papilla. Here you can appreciate more the mucosa, connective tissue in mucosa, the cells of connective tissue in the mucosa, and then the skeletal muscles. You can see peripheral nuclei. You can appreciate the peripheral nuclei. No nucleus is present in between the fiber, and here you can see the nuclei lying longitude. The nuclei, they are lying outside to the periphery of the cell cytoplasm, and this is the longitudinal fiber, and this is the cut section of the skeletal muscle fiber, and this is the lamina propria, and you can appreciate very well this is the blood vessel shown in the lamina propria. Okay. Now this is again the circumvallate papilla. So this is an actual picture of a patient and showing the circumvallate papilla. So these circumvallate papillas are present on the junction of the anterior 1/3 and posterior anterior 2/3 and posterior 1/3. They are present at the junction, as I have shown you before, and these are visible with the naked eye, and they have got a special structure. They are rounded structures and having space, a trench like space around them, and in this space on the lateral side of the papilla lies the taste buds. I can show you. These are the foliate papillas. You can see the taste buds lying on the lateral surfaces, and these are foliate papillas. Now this is the circumvallate papilla, and you can see various taste buds present on its lateral surface, and this is the trench which is present around the circumvallate papilla, and in this trench opens up some glands which are serous in nature, and these glands are known as von Ebner's glands. So the secretion of these glands provide a continuous flow of fluid over the taste buds that are abundant on the sides of these papillas and washing away food particles so that the taste buds can receive and process new gustatory stimuli. So these are the circumvallate papillas, and associated with circumvallate papillas are the von Ebner's serous glands, which secrete fluid into the trench-like space around the circumvallate papillas. Now this is the taste bud. You can appreciate the structure of taste bud, which is present in the epithelium of the fungiform papilla. So these are the taste bud. So these are ovoid structures within the stratified epithelium on the tongue surface, and which sample the general chemical composition of the ingested material. Now approximately 250 taste buds are present on the lateral surface of each circumvallate papilla, which may with many other present on the fungiform and foliate, but not on the keratinized filiform papillas, as I have told you before. They are not restricted to papilla, and they are also widely scattered elsewhere on the dorsal and lateral surfaces of the tongue, and where they are continuously flushed by numerous salivary glands. Now a taste bud these are ovoid structures and has got 50 to 100 cells, about half of which are elongated gustatory cells, which turn over within a 7 to 10 days span. Other cells present are slender supportive cells. So gustatory cells are the neurons which sense the taste, and the other supportive cells, they are present known as sustentacular cells, which support the function of these bipolar neurons. Also present are the immature cells and the slowly dividing basal stem cells that give rise to other cell types. Now the base of each bud rests on the basal lamina and is entered by afferent sensory axons that form these synapse with the gustatory cells. So here you can see the gustatory nerves are entering into the base of each bud and synapsing with the sensory neurons. At the apical ends of each gustatory cell, microvilli project towards a 2 micrometer wide opening in the structure called the taste pore. Now molecules, tastants dissolved in saliva contact the microvilli through the pore and interact with the cell surface taste receptors. So here the function of microvilli is to give surface area for taste receptors. Taste buds detect at least 5 broad categories of tastants. So this is the sodium ions, salty, hydrogen ions from the acid sour, and sugars and related compounds sweet, alkaloids and certain toxins bitter, and amino acids such as glutamate and aspartate umami flavor, umami savory flavor. This is the Japanese term. Salt and sour tastes are produced by ion channels, and the other 3 taste categories are mediated by G protein coupled receptors. Now, so this is the taste bud. You can see here some of the bipolar cells and some of the supporting cells and basal cells not shown here. So 3 types of cells are present in the taste buds. The bipolar neurons, which act as receptors for the taste, and the sustentacular cells, which are supporting cells, and the basal cells, which are stem cells, which act as stem cells. Now can you tell me the name of the nerve, which fibers are coming to this taste buds? Do you know it? Please tell me in the comments section if you know it. Okay, so now The taste bud, the posterior 1/3 of the tongue. So far we have covered the anterior 1/3 of the tongue. So now we will do the posterior 1/3 of the tongue. And in the posterior 1/3 of the tongue, I have told you that the posterior 1/3 of tongue is separated from the anterior 1/3 by the anterior 2/3 by a depression or a sulcus terminalis. The posterior region of tongue is located behind the circumvallate papilla and near the lingual tonsils. The dorsal surface of the posterior region typically exhibits large mucosal ridges and elevations or folds. So you can see the large ridges or elevations or folds, and that resemble the large fungiform papilla of the anterior tongue. A stratified squamous epithelium without keratinization covers the mucosal ridges and folds. The filiform and fungiform papilla that are normally seen in the anterior region of the tongue are absent from the posterior tongue. Lymphatic nodules of the lingual tonsils can be seen in the folds. I will show you in the next diagram a very good picture of it. The lamina propria, it contains of the mucosa is wider but similar to that of the anterior 2/3 of the tongue. Under the stratified squamous epithelium, this is the stratified squamous epithelium, and are seen aggregates of lymphoid diffuse lymphatic tissue and accumulation of adipose tissue, nerve fibers, blood vessels. Now, deep in the connective tissue of lamina propria and between interlacing skeletal muscle fibers are found the mucous acini of posterior lingual glands. Now, excretory duct, they open up into the surface of the tongue, and they provide lubrication for the passing bolus of the food. So, these are the glands which are opening up on the surface. So this is the lingual tonsils, that are aggregates of small individual tonsils, each with its own tonsillar crypt. And lingual tonsils are situated on the dorsal surface of the posterior region or the root of the tongue. A non-keratinized stratified squamous epithelium lines the tonsils and their crypts. You can see here non-keratinized epithelium, and they are located in lamina propria below the stratified squamous epithelium. Dense lymphatic infiltration surrounds the individual lymphatic nodule. You can see these are the individual lymphatic nodule, and dense infiltrate of lymphocytes are present around it. So this is the posterior 1/3 of the tongue. Okay, now this is the picture which you have to draw in your histology copies in the lab. Now you can appreciate the ventral surface. The ventral surface has got projections, various projections, and in them the filiform projections are more as compared to the fungiform, and the circumvallate are not shown here because they are present posteriorly. And you can see below the epithelium lies the lamina propria, and then the muscular tissue, and muscles are running in different directions. So we have the sections, and again then the lamina propria, and then the ventral surface of the tongue representing the now this is represented by the epithelium, smooth epithelium, stratified squamous, non-keratinized epithelium, and underlined lamina propria. So this is the picture you will draw in your copies, and in another video I will show how to make this diagram very easily. This is very easy, looking very tough, but it is very easy to make. Now I hope you this video has you understand you have understand the structure of the tongue by this video, and in the last the two identification points, the important identification points by which you identify the structure of the tongue is the presence of papillas, papillas on the dorsal surface and skeletal muscles and presence of the taste buds in the fungiform papillas. Okay, thank you. I hope this video is useful for you. Thank you.